Tactile stimulation during development alters behavior and neuroanatomical organization of normal rats, by Chelsea Merritt and Stephanie Bray.
The tactile stimulus mimics mothers licking and grooming their newborn rats, which is considered a positive experience. The tactile stimulus was applied 3 times daily to newborn rats from postnatal day 3 to postnatal day 21. The control group did not get any type of stimulus. When the rats are adults, we observed them performing 4 different tasks and compared them to the control group. The tasks consisted of the Wishaw tray reaching task, where the rat's ability to reach and obtain food was recorded. The tasks also included the activity box and the novel object recognition, where they chose between an old familiar object or a new familiar object. The tasks also consisted of the elevated plus maze, which is shown in the picture above. We then euthanized both the tactile stimulus group and group with no tactile stimulus. We measured dendritic length, branching, and spine density in the CG3 and AID areas of the prefrontal cortex and in the amygdala and compared.
Many previous experiments have examined negative early life experiences on brain development and neuroanatomy, but very little experiments have observed positive early experience. Because studies have shown that the brain is very plastic during development, we hypothesized that it should also change in response to enriching experiences like tactile stimulation. Evidence has shown that early environmental stimulation can greatly impact brain organization, cognitive ability, and motor abilities.
The mechanism underlying the effects of tactile stimulation are unknown, but it is hypothesized that tactile stimulation causes increase in production of a potent growth factor called FGF2. Because FGF2 is a growth factor, it is considered to be responsible for cortical plasticity. Researchers have previously examined effects on tactile stimulation on remediating brain injury, pain management and maturation for premature neonates, curing anxiety, and reducing drug sensitization.
Classic. Let's go. Taking the treatment in account, between the rats in the tactile stimulus group and the rats in the no tactile stimulus group, there were no sufficient differences in the time spent in the open arm or closed arm. The activity level of both of the groups also did not significantly differ. The sex of the rats also had no effect on the activity in both groups. Tactile stimulation had a significant effect on the performance of the Wishaw tray reaching task. However, there was no effect of sex on this performance. For the novel object recognition task, tactile stimulation had a very significant effect. The tactile stimulation rats preferred the new familiar object, while the no tactile stimulation rats preferred the old familiar object. In this case, there was also no correlation between sex and performance.
Synaptic organization in the prefrontal cortex and amygdala was greatly affected by the tactile stimulation. Dendritic branching, length, and spine density all increased in both sections of the prefrontal cortex. The tactile stimulus increased spine density in the amygdala. Sex only affected the spine density measurement of the amygdala.
We formed 3 ideas from our study. 1. Tactile stimulation alters certain behaviors in normally developing rats. 2. Normal brains exhibit dramatic alterations to dendritic morphology and organization in prefrontal cortex and the amygdala exposed to early tactile stimulation. Third, neuroanatomical changes associated with tactile stimulation are not sex dependent. The morphological changes in both the male and female's brains were the same, except for the spine density in the amygdala. A previous study showed tactile stimulation facilitated maturation of the visual system in both human infants and rat pups, and increased activity on an EEG, a device that measures electrical activity on the scalp. This is consistent with the idea that tactile stimulation alters cerebral development and shows that it is possible to enhance motor functions, as demonstrated by the tactile stimulation group during the Wishaw tray task. Based on past findings, we hypothesized tactile stimulation would decrease the adult rat's anxiety levels, and they would spend more time on the open arms of the elevated maze. This was not the case, however. We believe that the no tactile stimulation group, the control group, may have lowered anxiety levels due to 3 previous experiments. The tactile stimulation group preferred the new familiar object. This is opposite of what we hypothesized, because usually better scores in the novel object recognition task are associated with preferring the old object. We think this may be because many of the tasks are designed to measure adverse experience effects and not enriching ones.
Researchers have previously shown the effects of electrophysiological measures of brain development, but this study is the first to demonstrate the widespread effects of tactile stimulation on synaptic organization in the prefrontal cortex. Little previous research on neural organization after tactile stimulation exists. One past study found 14 days of tactile stimulation early in rats' life showed decreases in spine density and dendritic length in the parietal regions. However, we found significant increases in spine density, dendritic branching, and length in the prefrontal cortex. We believe tactile stimulation may affect different brain regions in different ways. If this is true, then our different findings could be because tactile stimulation enhances FGF2 expression in the skin and prefrontal cortex.
This study proves that just like early adversity, tactile stimulation during a critical developmental period has the ability to dramatically reorganize dendrites in multiple brain areas and influence adult behavior. Therefore, tactile stimulation is a powerful brain reorganization tool and could play an important remedial or preventative role in a variety of neurodevelopmental disorders. Because tactile stimulation greatly increases connectivity between surrounding neurons through physical amplification, mothers can perform tactile stimulation to increase the intelligence of their child. Longer dendritic length, increased branching, and larger spine density can cause signals to become quicker and more sufficiently spread, resulting in increased memory skills. The gratifying success of the Wishaw tray reaching task shows that tactile stimulation can greatly improve motor function.
There are 2 things that we would have changed about this experiment. Number 1, instead of having one tactile stimulation group do all 4 tasks, we would have 4 tactile stimulation groups and 4 non-tactile stimulation groups, all being raised the same way, but each only doing one of the tasks. That way, there would be no question about the rats getting more comfortable as they perform each task. Number 2, we would change the novel object recognition task to something that measures an actual benefit to tactile stimulation.
Go do another one.